Hi everyone, welcome back. It's been a long time since my last video and I guess it's going to change now for good. So let's begin. Today we are going to discuss about the physiology of vitamin D. Today I am going to discuss about the physiology of vitamin D. Vitamin D comes in many forms. In humans, two forms are found. One is called vitamin D2 and another one is vitamin D3. D2 is also known as argocalciferol and vitamin D3 is known as cholecalciferol. Before I go any further, it is good to know that vitamin D is a vitamin as well as a hormone. But why is it so? I will give you the answer before the end of this video. So stay tuned till the end of the video. Whether a hormone or a vitamin, the question is, how can we get them? Well, we can get vitamin D in diet or we can synthesize them. But as I told you, there are two forms of vitamin D present in human body, D2 and D3. And there are some differences between the two. That is the difference in terms of their sources. As far as vitamin D3 is concerned, the good thing is we can get enough of vitamin D3 without having to get them from diet. That is, we can synthesize them. If our skin is exposed to adequate sunlight so that adequate amount of UVB can be absorbed, 7-dehydrocholesterol in skin is converted into cholecalciferol or vitamin D3. The dietary source of vitamin D3 is mostly from animal sources such as cod liver and halibut liver, egg yolk, fortified milk, etc. But the story is not the same for vitamin D2. D2 must come entirely from diet and diet of plant sources only. So to summarize, there are two forms, D2 and D3. D2 comes from dietary sources only, but D3 can be synthesized as well in addition to dietary sources. You know, vitamin D is a fat-soluble vitamin. So, for its absorption, we need bile salts so that they can be emulsified and can be absorbed. Once absorbed, it is carried in blood in chylomicron or it may be bound to vitamin D binding protein. Vitamin D, either D2 or D3, they are not biologically active. So to exert their action, they must be converted into some biologically active metabolite. What are they? Well, these are the active forms: 1,25-dihydroxyvitamin D2 and 1,25-dihydroxyvitamin D3, also known as calcitriol. They are the most active form of the vitamin. You can see I have underlined the word most, and this is purposeful, and you'll get to know soon. Now let us discuss how vitamin D is converted into its active metabolites. Whenever the 25-hydroxyvitamin D forms are low in concentration in the blood, the adipose tissue releases vitamin D in blood. Once released into the blood, vitamin D2 or D3 are converted into their respective 25-hydroxy forms, that is 25-hydroxyvitamin D2 or 25-hydroxyvitamin D3, and this conversion takes place in liver. The enzyme 25-hydroxylase, which facilitates this reaction, is a member of cytochrome P450 family. The 25-hydroxy forms are still not the most active forms. The next conversion into the dihydroxy forms, which is the most active form of vitamin D occurs in the kidneys. In the proximal convoluted cells of nephrons, another enzyme, 1-alpha hydroxylase, which also belongs to the same cytochrome P450 family of protein, converts the 25 hydroxyforms into 125 dihydroxyforms, the most active metabolite of vitamin D. As I mentioned previously, the dihydroxyforms of vitamin D is the most active form of the vitamin. This is because they bind to their receptors with an affinity that is about 1000 times more than the monohydroxy forms, that is the 25 hydroxy forms. But again, in terms of concentrations in which they are present in blood, the 25 hydroxy forms are present at a concentration about 1000 times that of 125 dihydroxy forms. So, though 125 dihydroxy forms are the most active form of the vitamin, both of them are probably biologically active. From the discussion so far, it is clear that the vitamin D in their original form, that is the argocalciferol or cholecalciferol, is converted first into 25 hydroxy forms in the liver with the help of 25 hydroxylase, which is a member of cytochrome P450 group of enzymes, and then again it is converted into 125 dihydroxy form in the proximal convoluted tubule of nephrons by the enzyme 1-alpha hydroxylase, which is again a member of cytochrome P450 enzymes. This is how the active metabolite is produced, that is the most active metabolite. The first conversion from argocalciferol or cholecalciferol to 25 hydroxy form, which occurs in liver, is not regulated. The second conversion from 25 hydroxy forms to 125 dihydroxy forms is highly regulated. And here is a summary of how the regulation plays out. You can see that active metabolite, the product of the reaction, negatively feedbacks the 1-alpha hydroxylase enzyme, thereby inhibiting its own formation. On the other hand, PTase, which increases when plasma calcium concentration is low, provides positive feedback to the enzyme, promoting conversion of 25 hydroxy form into 125 dihydroxy forms, that is the most active metabolite. Another factor involved here is fibroblast growth factor 23, FGF23. It is released from osteocytes and osteoblasts, and it is released in response to high plasma concentration of phosphates. Because one of the actions of the active metabolite of vitamin D is to increase phosphate absorption and thereby increasing phosphate concentration in blood, it's quite natural that phosphate has some way of negatively feedbacking the production of active metabolite of vitamin D, and this occurs through the action of FGF, that is fibroblast growth factor 23, released from osteocytes or osteoblasts. Now that we know what vitamin D is, how we can get them, where we can get them, and how they're metabolized in the body, let's dive into its actions. In this video, I shall discuss on 3 of the actions of vitamin D, that is its actions on small intestine, on nephron, and on bones. So let's begin. The main function of vitamin D in small intestines is to increase calcium and phosphate absorption. Let's discuss about the calcium absorption first. Calcium absorption in small intestine occurs through 2 pathways, the paracellular pathway and transcellular pathway. In the paracellular pathway, calcium passes through the in between of the cell through the cellular junctions, and this pathway is present all over the small intestine. Also, paracellular pathway is the major pathway for calcium absorption in the small intestine. Here is a representation of the paracellular pathway in the picture. You can see it. Once again, it's the major pathway, and the most interesting fact about it is that paracellular pathway, though the major pathway, is not regulated. And vitamin D plays no role in this pathway. The transcellular pathway is where vitamin D comes into play. Transcellular pathway means calcium must pass through the cell. That is, it will enter the cell through a receptor, then it will exit the cell through another receptor. One important feature of transcellular pathway is that it exists only in the duodenum, and vitamin D regulates this pathway only. So, how is it that vitamin D increases calcium absorption in the duodenum through the transcellular pathway? To understand that, we need to understand two more things. One is mechanism of action of vitamin D in general. It is applicable anywhere in the body. And second is mechanism of calcium transport in the duodenum. So first, let's discuss the mechanism of action of vitamin D. Vitamin D receptor is a nuclear receptor. It belongs to the nuclear receptor family. And the name of the receptor is very easy to remember, vitamin D receptor, that is VDR. When vitamin D binds its receptor VDR, this receptor dimerizes. It forms a heterodimer with retinoid X receptor, that is VDR binds to retinoid X receptor, and this complex then binds to a hormone response element, or in this case a vitamin D response element, on the 5' end of a gene of interest. And by binding to the DNA segment, it acts as a transcription factor. It may inhibit transcription, it may promote transcription, and thereby vitamin D exerts its action. Now that you know the mechanism of action of vitamin D, let's come back to an enterocyte of duodenum. Here you can see on the apical side, that is on the lumen side, a transporter is present, calcium transporter by the name TRPV6. The full name is transient receptor potential vanilloid type 6. Calcium enters into the enterocyte through this transporter, and calcium exits the enterocyte through two more transporters. One is sodium calcium exchanger and another one is a calcium pump. Both of them extrude calcium out of the enterocyte into the extracellular fluid and finally into blood. And there is another protein that helps in this process. And that protein is calbindin. What it does is it sequesters all the calcium in the present in the enterocyte, thereby decreasing intracellular calcium concentration, and thereby it helps to maintain the concentration gradient from outside to the inside of the cell. Well, I'm talking about all the transporters and proteins, but what's the role of vitamin D in it? And here you are. Vitamin D, through its action on VDR, it acts as transcription factor and promotes transcription of TRPV6, calbindin, and calcium pump, all the proteins necessary for transport of calcium from the lumen into the enterocyte and finally into bloodstream. And this is how Vitamin D promotes calcium absorption in the duodenum through transcellular pathway. Well, what about phosphate absorption? Phosphate enters the cell through sodium phosphate cotransporter, and this step is the rate limiting step for phosphate absorption. And what vitamin D does is it increases the synthesis of this transporter and thereby increases phosphate absorption in the intestine. Now it's quiz time. What do you think about this? Just pause the video for a moment, have a look at it, and then come back. Yes, it does have a role in calcium absorption in the small intestine, but PTase doesn't have any receptor there. So it acts through vitamin D. How? It positively feedbacks on 1-hydroxylase, the enzyme that is responsible for formation of active metabolite of vitamin D, which in turn increases calcium absorption in small intestine. So if you ask me, no, there is no direct role of PTase, but it acts indirectly through vitamin D, through production of vitamin D. What's the action of vitamin D on nephrons? Well, it's the same as that in small intestine. It increases calcium and phosphorus reabsorption. Just remember the site of action, that is DCT, where vitamin D acts on the nephron. Those of you who are familiar with PTase and its action on nephron, what do you think? Is vitamin D synergistic with PTase or is it antagonistic to PTH? As far as calcium reabsorption is involved, PTase is the main hormone responsible for this function. And not only that, PTase is the dominant hormone here. And in case of parathyroidectomy, calcium reabsorption in the nephron decreases, and vitamin D cannot compensate for the loss even with higher doses of vitamin D. Well, as far as phosphorus reabsorption is concerned, PTase causes phosphorus excretion, not reabsorption, and vitamin D causes reabsorption. So in this case they are antagonistic to each other, and again PTase is the dominant hormone. So far we have understood that all vitamin D does is absorption or reabsorption of calcium and phosphorus from small intestine and nephrons respectively. Quite simple, isn't it? But now comes the trickiest part of it. Actions of vitamin D on bones. I will try to make it as simple as possible, so stay tuned. Vitamin D acts on bones in two ways. It has some indirect actions and some direct actions. Let's first understand the easier one, that is the indirect action of vitamin D on bones, and thankfully enough, this is the major action of vitamin D on bones. The major mineral component of bones is hydroxyapatite, which is composed primarily of calcium and phosphate. Vitamin D, through its action of absorption and reabsorption of calcium and phosphate in the small intestine and nephron respectively, increases the total plasma concentration of calcium and phosphate. Thereby, it increases the availability of these two ions for mineralization in the bone. So, indirectly, vitamin D increases bone mineralization. And that's the indirect action and the major action. Now let's move on to the direct actions. You know that there are three types of cells in the bone: osteoclast, osteoblast, and osteocyte. Here we shall discuss about two of them only. Vitamin D has got receptors on osteoclast precursors and osteoblast precursors. Through its action on osteoclast precursor, it causes proliferation of osteoclasts, which are involved in bone resorption. And through its action on osteoblast precursors, it forms osteoblasts, which are the main bone-forming cells. So you can see at the same time it is activating both the osteoclast and osteoblasts. So when these actions are balanced, that is, osteoclastic and osteoblastic activities are balanced, there is no innate bone formation and no innate bone resorption. The only thing that is happening is increased bone turnover. Before any further discussion on vitamin D, let's understand the things that are written in red ink. Osteoblasts release some cytokines, rank ligand and macrophage colony stimulating factor. Both of them increases the proliferation and function of osteoclasts. So, if vitamin D remains active for a long time or vitamin D is in excess, what happens is that the balance is disturbed. That is, osteoclastic and osteoblastic activity is no longer going on at the same rate. What happens is, because osteoblasts through rank ligand and macrophage colony stimulating factor activates osteoclasts, continued action of vitamin D on osteoclast and osteoblast precursor will eventually shift the balance towards bone resorption. Because osteoblasts produce rank ligand and MCSF, which increases osteoclastic activity. This complex balancing act of bone resorption and bone formation influenced by vitamin D can have its uses in certain circumstances. Sometimes, some site may require bone formation at the cost of bone resorption at some other site. So, to summarize, on bones, vitamin D has got indirect and direct actions. Indirect action is the major action, which is bone forming action, that is mineralization action, because vitamin D makes available calcium and phosphate in the blood, and this bone mineralization is the main action of vitamin D, the major action. And when vitamin D is in excess, through direct action, it can cause bone resorption. One direct action I forgot to mention so far is that vitamin D stimulate osteoblasts to produce osteocalcin. Osteocalcin is a protein which helps in bone mineralization by acting as nucleation site for hydroxyapatite crystal deposition. Finally, let's connect everything together. That is, the two main hormones responsible for calcium homeostasis, PTH and vitamin D, the active metabolite of vitamin D. As discussed previously, active form of vitamin D exerts negative feedback on 1-alpha hydroxylase, thereby inhibiting its own production. Increased phosphate concentration in blood causes release of FGF23 from osteocytes and osteoblasts, which again inhibits 1-alpha hydroxylase and thereby preventing formation of the active metabolite of vitamin D. And vitamin D also has negative feedback on the chief cells of parathyroid gland. Here it decreases the synthesis of parathyroid hormone. Detailed discussion of calcium homeostasis is beyond the scope of this video, and that I might do on another day. So let's conclude with vitamin D deficiency, which is why we need vitamin D the most. Deficiency of vitamin D during childhood causes decreased availability of calcium and phosphate in blood, and thereby decreased mineralization of bone. Decreased mineralization of bone causes decrease in strength of the bone, which results in characteristic bowing of weight-bearing long bones in children. In adults, osteomalacia is the result of vitamin D deficiency. Because bone growth has already occurred, bowing of the bones, long bones, are not seen here, though microscopically the structure of the bone is similar to that found in ricket. Here also, new mineralization is not happening and osteoid amount increases. This leads to increased propensity for fracture. Microfractures are often present, which are difficult to detect radiographically, and both conditions are treatable with the help of vitamin D administration. That's it for today. See you in the next video soon.